A very good morning to everybody. I am Dr. B. Shanti, Professor and Head, Department of Biochemistry of Sree Balaji Medical College and Hospital, Bihar. And today will be the very first topic and the core basic of biochemistry, that is the cell, which comes under the competency 1.1. And as you all are very aware, the whole of the biochemical processes inside the body are happening inside the cell and also into designated subcellular organelles. Each subcellular organelle plus the cell membrane has got a specific function and very beautifully compartmentalized function. So it will be a real glory to walk along with the understanding of the cell and the performance as a cell. So in another rather half an hour, you'll be sailing along the journey with me to understand the intricacies of the cellular performances and the beauty of the subcellular organelle. Under competency 1.1, the specific learning objectives of the topic cell will be description of the basic structure of the cell, enumeration of the functions of the various subcellular organelles, explanation of the structure and function of cell membrane. Now, cell is the structural and functional unit of life. The cell contains various organized structures collectively called as cell organelles. So this is the structure depicted of the cell. So now we will be dealing with the important organelle of the cell, which is the nucleus. Nucleus is a very prominent organelle of the cell. All cell, every cell contains nucleus except mature RBC and uppermost layer of the skin. In some cells which are of very great functional significance, the nucleus occupies almost most of the available cellular space. If the cell is this big, 3/4 of the cell space will be occupied by the nucleus itself. For example, lymphocyte and spermatozoa. The nucleus is surrounded by two membranes. One is the inner membrane, which is called as the perinuclear membrane, which has got numerous pores. Why pores? Allow the entry and exit of substances, which is very important. Outer membrane, and the outer membrane is continuous with the membrane of the endoplasmic reticulum. So now nucleus. Nucleus again, we'll continue. Nucleus contains a very, very vital substance called deoxyribonucleic acid or DNA. This DNA is the chemical basis of the genes, and the genes govern all the functions of the cell. Very long DNAs are complexed with proteins. When they are complexed with proteins, they are called chromatins. And further, these chromatins will organize themselves into chromosomes. The DNA replication and RNA synthesis. DNA replication means the division of a DNA. Once DNA divided into multiple 2 DNAs. Likewise, RNA synthesis, that is transcription, takes place inside the nucleus. A lighter shaded area in the nucleus is called as nucleolus, and that is where RNA processing will take place. So DNA replication and RNA synthesis, and here the structure tells you all. Got it? Okay. Now we'll go to the structure, another subcellular organelle called Golgi apparatus. This was first described by a person called Camillo Golgi. How does Golgi apparatus look like? That is a network of flattened smooth membrane and vesicles. While moving through the endoplasmic reticulum, carbohydrates are attached to proteins, nascent proteins. When carbohydrates are attached to proteins, what are they called? They are called glycoproteins. And while moving through the endoplasmic reticulum, carbohydrates are getting attached to proteins and they are called glycoproteins, and they reach the Golgi apparatus. See? So this structure tells it. Golgi apparatus, flattened smoothened membranes, vesicles, and glycoproteins traverse from endoplasmic reticulum to the Golgi apparatus. So now, as the picture showed, they are formed into secretory vesicles. When they are formed into secretory vesicles only, they can be stored and sent to the respective destination. So they are formed into secretory vesicles where the finished products, that means glycoproteins, are stored, lipoproteins, glycoproteins, etc., are stored and released when there is an appropriate stimuli or when it is needed. So the functions of the Golgi apparatus include protein sorting, protein packaging, and protein secretion. So the Golgi apparatus says it all. Protein sorting, packaging, and secretion. Now we'll go to endoplasmic reticulum. Once again, this endoplasmic reticulum is an interconnecting membrane with channels and cisternae. So now this is very prominent in cells actively synthesizing proteins, because the function is, there are two types of endoplasmic reticulum which is actively involved in protein synthesis. So this endoplasmic reticulum will be prominently present in cells which are actively synthesizing proteins. What are these proteins which are synthesized in endoplasmic reticulum? Immunoglobulins secreting plasma cells, glycoproteins, lipoproteins are all formed in the endoplasmic reticulum and targeted to the Golgi apparatus. So that is about endoplasmic reticulum. Very prominent in cells. Okay. Now the functions of endoplasmic reticulum is one is detoxification of drugs. Okay, how many types of endoplasmic reticulums are there? There are two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. What is the difference? The rough endoplasmic reticulum has got ribosomes attached. Why? What is the function of ribosome? Protein synthesis. So we know why rough endoplasmic reticulum is more predominantly present in cells where protein is synthesized. So now we go about another organelle called lysosomes. These are very tiny organelles. It's a bag of enzymes. Now what is the function of enzymes? To break down. So this is a bag of enzyme and it is also called a suicidal bag. Why is it called a suicidal bag? Because it has enzyme which will be destroying all the macromolecules after their function is achieved. So what is the bag of enzymes and what are their functions in lysosome? Glucosidase and galactosidase. They will be hydrolyzing polysaccharides or carbohydrates. Cathepsin collagenase are examples of enzymes which will hydrolyze protein. Elastase and peptidase are again protein hydrolyzing enzymes. Nucleic acid hydrolyzing enzymes like ribonuclease and deoxyribonuclease are also present in the lysosomes. By what enzymes lipid are hydrolyzed? Fatty acid esterases and phospholipases. So you get the function of lysosomes, which are again called suicidal bags, and they contain enzymes which will hydrolyze the macromolecules. So this is a lysozyme. Single-walled structure. Enzyme complexes are present inside the lysosome, and it will break all the, or rather hydrolyze all the macromolecules with the help of the respective enzymes. Tiny organelles, bag of enzymes. So protein hydrolyzing, lipid hydrolyzing, nucleic acid hydrolyzing. It has got a granular matrix. Oh, now we talk about peroxisomes. This is another cellular organelle. This has got a granular matrix, and it is very prominently present in WBCs, leukocytes and platelets. They contain enzymes like peroxidase and catalase. So the functions of the enzymes is to destroy free radical formed by peroxidation of polyunsaturated fatty acids. See, peroxisomes. They are granular matrix, peroxisomes, single membrane. They contain what? Antioxidant enzymes. So they have antioxidant properties. They contain enzymes like peroxidase and catalase. Granular matrix found in leukocytes and platelets. The absence of these peroxisomes can lead to some of the disorders like adrenal leukodystrophy, and it will lead to insufficient oxidation of fatty acids, and another syndrome called Zellweger syndrome due to peroxisomal disorder. Adrenal leukodystrophy and Zellweger syndrome. Okay, now we talk about one important organ called mitochondria. Everybody should be aware of this mitochondria. They are spherical, oval or rod-shaped bodies. Erythrocytes or the common RBCs lack mitochondria. They don't have mitochondria. Whereas the tails of spermatozoa are ridden with mitochondria, full of mitochondria. What is the function of mitochondria? It's a powerhouse of cell, and it enables oxidation of foodstuffs. And what is the energy trapped? It is called as the powerhouse of cell. Powerhouse of cell means what? It contains the energy. And what is that energy? That is a chemical energy called ATP. Now the mitochondria has two membranes. One is the outer membrane and the other one is the intermembrane. See, this is the structure of mitochondria. Outer membrane and inner membrane and cristae and inner matrix. Powerhouse of cell. Spherical, erythrocytes no mitochondria, spermatozoa full of mitochondria, powerhouse of cell, and outer membrane and inner membrane are present. Inner membrane, as you saw in the picture, folds into cristae. And what are the functions of this? How does the function of the mitochondria is enabled? All the enzymes of the electron transport chain, citric acid cycle, urea cycle, heme synthesis are present in the mitochondria. Cytochrome P450 is present in the inner mitochondrial membrane. Antioxidant enzymes are present in the cytosol of mitochondria called superoxide dismutase. Mitochondria contains a DNA called, we saw that nucleus contains DNA. Mitochondria nowadays also, so much of importance and focus is being given on this, which is called as mitochondrial DNA. So mitochondria. Now we know the functions of mitochondria and what are all present in the mitochondria to enable that function. Okay, fine. Now we know every organelle has got one function, and there are certain marker enzymes which will identify that particular organelle. What are the marker enzymes? We all know mitochondria is the powerhouse, and so ATP synthase is the marker enzyme. Lysosome is the suicidal bag and enzymes breaking down, and it is marker enzyme is cathepsin. Golgi complex, where all protein sorting and packaging and glycoprotein synthesis take place, and that galactosyl transferase is the marker enzyme. Microsomes, glucose 6-phosphatase. Cytoplasm, lactose dehydrogenase are the marker enzymes of the subcellular organelles. Now plasma membrane. The membrane separates the cell from external environment. It's selectively permeable. Selectively permeable means It's only certain substances can be allowed in and out. Not like every substance can freely enter and out. Only whatever required substances at that point of time will be allowed to pass across the membrane. That is regulated. What is a membrane made up of? It is made up of lipids, proteins and carbohydrates. That means glycoproteins and glycolipids. See the structure of the membrane: carbohydrates, glycoprotein, protein and lipids. So hydrophobic means it does not allow water. Hydrophilic means allows water. See, hydrophobic and hydrophilic. This is called fluid mosaic model of the plasma membrane, bilayered membrane. So selectively permeable, lipids, proteins and carbohydrates. And what are the types of lipids? The lipid which is commonly constituting a plasma membrane is a phospholipid. Okay, and it is amphipathic. Amphipathic means, I told you, hydrophilic and hydrophobic. It can allow, one side it can allow water and one side it will not allow water. Cell membranes also contain cholesterol. Some enzymes are also present in the plasma membrane. Those enzymes are alkaline phosphatase and 5' nucleotidase. They are called exoenzymes, ectoenzymes. So plasma membrane, you saw, this is fluid mosaic model, and inner hydrophobic and outer hydrophilic membrane. Cholesterol and phospholipids are the constituent of the plasma membrane and they are selectively permeable. Okay. Ectoenzymes are present on the external side of the membrane, that is outer part of the membrane. So it is semi-permeable. So it can allow certain functions, certain substances to pass in and out of the membrane. Only regulated traffic, that's called selectively permeable. Unwanted molecules, spinner. What are the various types of transport across the plasma membranes? Passive transport, active transport, pump, simple diffusion, facilitated diffusion and ion channels. Now we'll go one by one, rapid rush. So most of it will be dealt with in other sessions. So I'll just introduce you to the types of diffusion, transport. Active transport. Active means you require energy. So that says that. So the active transport mechanism requires energy. Unidirectional. That means it can transport substances only in one direction. And it requires integral proteins. Only in the presence of proteins that kind of a transport can take place. Active transport can take place only if there are integral proteins. So one-sided, requires energy and they are susceptible to inhibition. It can be inhibited. The transport, active transport can be inhibited by certain compounds. Sodium pump, calcium pumps are all examples of active transport. See, active transport here. Energy, ATP is there and so one direction only that's taking place, susceptible to inhibition and pump. Energy, integral proteins are needed, susceptible to inhibition and example. Passive transport. So, as the name suggests, it does not require energy. Simple diffusion. See, concentration gradient. That means normally where there is more concentration, anything tends to flow towards lower concentration. So since it is driven by a concentration gradient and since the movement of the substances or solutes is from higher to lower concentration, no energy is required. And here in simple diffusion, there will be no carrier is required. Whereas in facilitated diffusion, there will be some carrier is required to carry. Though it is from higher to lower concentration, some carrier is needed to take that solute across the membrane. This diffusion is, this can operate bidirectionally. Whereas you saw in active transport, it was only in one direction. Whereas in diffusion, it can take place bidirectionally. What are ion channels? They are proteins. Transmembrane, present in the membrane. These proteins are transmembrane proteins. That means they are present in the membranes that allow the entry of ions. What are ions? Ions like sodium, potassium, calcium and chloride. So for the transport of substances like sodium and potassium and calcium, you have special channels called ion channels, which are nothing but transmembrane proteins. Okay. Passive transport. See what is happening? Simple diffusion. So no energy is required from higher concentration, lipid bilayer enzyme, membrane from higher to lower. Ion channel, sodium, calcium pump is transported across. See, facilitated diffusion is a carrier is present here. Now active transport energy is required. So this picture says it all. Transport systems are classified as uniport, symport and antiport. The name is suggests uniport means only one solute will go. Symport means two solutes in the same direction. Antiport means two solute, one in one direction and the other one in the opposite direction. See, uniport carries single solute across the membrane. Glucose transporter, example. Symport carries two solutes in the same direction. That is sodium dependent glucose. So two solutes like sodium and glucose are transported, and that is transported in the same direction. Antiport means two solutes are transported, one in one direction and the other one in the opposite direction. That is sodium potassium transporter. So this is all about the transport mechanisms across the bilayer plasma membrane. Okay, so this is again what it says. Uniporter is single, symporter is double in the same direction, antiporter in the opposite direction. So today we have successfully dealt with various compartmentalized subcell and subcellular organelles and their distinctive functions with their respective marker enzymes and activity, and how these cellular organelles can be separated also. And we also dealt with about the cell membrane, plasma membrane and various transport mechanisms through which the solutes can be transported in and out of the various membranes. So the definite take-home message is you'll have to know the distinctive functions of the cell in their respective tissues, and also the transport mechanisms which enable the transport of important solutes and solvents, and the mechanisms of transport across the membranes. Thank you.